Thank you for that amazing, kind introduction. Indeed, I joined really recently, but I'm happy to share today the whole knowledge that I have regarding this long COVID project that we are doing in Protobios and share the preliminary results that we have in our hands. But first, would be happy to talk a little bit about the Protobios where I'm working right now. So I was looking for a new challenge in my life, and I found that this challenge in Protobios, because it's amazing biotechnology company that is private company that is focusing on the development of high-tech clinical tools. And indeed, we've been working in the Protobios have been working for many years to develop this MVA technology that we will discuss later. That is now patented, and therefore we are participating in different programs to help people, to help research institutions, researchers to see the immunoprofiles of the patients and to create a different following analysis regarding the immune system of those patients. So what we are doing basically in simple words that we are supporting without things, without the research that we are doing inside our building is inside our house is that we're supporting biomarker testing, immunoassay development, and different projects and different diseases. And our major goal is that the major goal that we're trying to achieve is that we are dreaming about the improvement of the quality of life through the basically 3 major ideas, steps, and call it whatever you want. So we really hope that our technique that we've been using also in this project is actually can help in assessment of the drug efficacy. So in other words, it helps to evaluate the effectiveness of therapeutic interventions, and it offering insights how the treatment can modulate the host immune responses. In very simple words, we can actually we could be able to distinguish between healthy and not healthy, but between responders and not responders to the treatment. That was the goal that we are trying to reach with our technology. We also want to contribute to the disease prevention, to identify specific immune signatures through which we can predict the occurrence of different diseases. And also we are trying to optimize the clinical trials for antibody therapeutics, antibody studies, because again, this is very important to understand what kind of people can be recruited for different clinical studies. And with the immunoprofiling, we can also say that whether this person will react to this treatment or maybe not, or maybe any other subgroups we can modify. So we can improve the quality of the clinic, not just the clinical study, but also the quality of the test for the participants who are participating in this clinical study. So this is the things we're trying to achieve with our technology and what we are doing in our labs. And now I'm going to talk about our famous MVA technology, mimetic variation analysis. This is how we analyze the immune system. And I'm going to create a complete mess to you because I'm going to tell you that we are using the bacteries, modified bacteries, vector-fags, actually, to analyze the immune system and to see the viral effect, the effect of the viruses to the immune system, of different viruses, of new viruses, of the viruses that already persist in our bloodstream. So to simplify, this is the method at the end of the day, this is the method that helps us to distinguish between 2 groups: healthy individuals regarding this disease and affected individuals regarding this disease. This is one of the first steps that helps us to do. So the idea is that we are generating synthetic immune repertoire which consists of billions of different types of molecules. And we are creating the peptide antigens, and those peptides that we call mimotopes, they are designed to mimic the epitopes of natural antigens that the immune system might encounter. And when antibodies from a biological sample like blood or are exposed to this diverse library, so specific binding interaction occurs inside the sample, blood sample, and this allows us to identify this kind of recognize those antigens and those mimotopes to which the individual's antibody can bind. And therefore, we can see the structure of the immune system that is occurring at the same moment, like we've been taking the sample. So the critical aspect of this MVA is that we compare always case and control. So case is usually refers to the individuals with a specific disease, controls is the healthy individuals groups. And by comparing those patterns between the healthy and unhealthy individuals, we can identify differential antibody responses and see and conclude the next things and prepare the next analysis to distinguish even for more patterns. And here's the example how the MVA actually works. So this is the broad example that, for example, we are taking the human, we are taking his blood sample, and we start to analyze his antibody. We are extracting antibodies from his blood sample or her blood sample. And next, we are capturing those antibodies with our library that contains like billions of molecules, and then we can cluster that with the different algorithms that we also constantly developing. This is mostly ML algorithms or AI algorithms that helps us to go very deep into these patterns. And we see different patterns indeed here, like in the blood sample, while some of them you can see here, they are very specific, for example, for cytomegalovirus that is constantly presented in our body. Or, for example, we can see that here's something happening with the Helicobacter pylori infection in this patient at this moment. And going to the next step, when we are getting even closer to that, into these patterns, we actually can distinguish that, okay, we can see some specific epitopes or mimotopes that are specific for different diseases, actually. For example, for multiple sclerosis, some bunch of here of epitopes, or for Parkinson disease, a couple of clusters here, or a lot of the samples that we clusters that we hear specific for psychosis associated first episode of psychosis that can be associated with Parkinson or schizophrenia. So for us, seeing this pattern, we can actually conclude for this particular person, we can, if this person will develop the first signs of neuro disease, we can say that probably it's because of the Parkinson. So it's like, it's much deeper analysis, but it's just an example how we can distinguish based on the immune system and how deep we can go with profiling of the immune system. And this is the published study just to show you the also like idea in the real examples. This was like just a broad example. This is the real example of the multiple sclerosis, which is slightly similar, a little bit similar to long COVID, because multiple sclerosis often starts with progress from the optical neuritis. So we know that those people with multiple sclerosis, 20% of them have the optical neuritis in the beginning. But not all the people develop the multiple sclerosis from the optic neuritis. So we decided to see whether we see kind of interesting immune patterns to be able to distinguish those patients with such diseases. And indeed, we found them, and which is most like mind-blowing, it's all associated also with another type of viruses that are sitting in our immune system, like in our bodies constantly. So in this case, we had cytomegalovirus and Epstein-Barr virus. Remember this guy. We will talk about this guy later in the context of the long COVID. So the idea is that something is happening to our body that causes the reactivation of Epstein-Barr virus or cytomegalovirus that causes the development of the optic neuritis into the multiple sclerosis. So what we found several specific epitopes that can actually be a biomarker for that kind of disease. And I'm not going to stop here for a long time, but just an example. This is how we can distinguish and understand what is the difference between different multiple sclerosis patients and how we can help to clinicians to explain to them also that this is why probably some medications can not be that effective or any other things. And let's talk about the long COVID. I personally hate COVID, honestly, because I struggled with COVID in last month. And some of the symptoms that usually people have for the long COVID, I also had them, and it's not a pleasure. So I'm really hoping for this project that we can help somehow to those people who are suffering from long COVID, because it's one of the most terrible diseases, like from the perspective, also from scientific perspective, because it has over 200 different ways how it can be manifested. It can be problems with heart, it can be problems with fatigue, it can be problems with memory loss or hair loss, or lots of different like smells or changes in the way how we can taste the food. Like thousands, like thousands, like hundreds, like different ways how we can understand that, okay, this is the long COVID. So we understand that if the pattern is so heterogenic, this means that there are so many things behind this disease actually that can influence, that can affect this appearance of this disease. And it will be a very hard, big challenge to us to hold the whole community to understand all these patterns. And here we started to study long COVID, and what we found that it's not only about the one virus, that SARS-CoV-2, it's also about the Epstein-Barr virus that is sitting inside us. So, yep, now we are talking about two viruses, actually. Sorry, I'll drink a little bit of water. Like our observations, in our observations, we see that we see elevated markers of ABV virus reactivation in a huge proportion of the data set of our samples and of our individuals. Mostly ABV is that virus is presenting in over 90% of the population. So during our life, we see that we are contaminated with that virus, like infected with this virus, we are getting that virus anyway. For most of us, it comes without any symptoms. For some of the people, mononucleosis can develop, but like it's a common thing between all of us. And what we see is that initial SARS-CoV-2 reaction actually might be a trigger for the ABV reactivation and subsequent proliferation in certain individuals. And we still do not know the precise mechanism. We're still in the beginning of that investigation, but our hypothesis posits that dysregulated or overly heightened immune response during acute COVID-19 could compromise the host ability to maintain the EBV, which is already in latent state. So, yep, the idea is that our immune system is bringing all the resources to fight with the COVID-19. And during this time, EBV is waking up and starting his own damage that altogether, combined with some other conditions, brings to the long COVID syndrome. So think I said here everything I want to say here. So what we also see, unfortunately, the phenomenon of viral reactivation of long COVID is not limited only to Epstein-Barr virus. We also see that it may be associated with the reactivation of other viruses that sitting inside us, like cytomegalovirus and herpes simplex virus, that actually can prolong the persistence of the long COVID symptoms and the overall severity of the conditions. So this suggests a broader pattern of impaired immune surveillance over this latent virus infection after this SARS-CoV-2 infection getting in our body, and we are trying to understand how to distinguish between those things and what kind of factors can be involved also as well. As you know, some people have chronic diseases, and yes, they also can play their role. So everything is connected. It's a complete madness. So I'll try to explain in a very simple way these plots that we have here. So this is the visual representation that illustrates this concept by showing the effect size of antibody activity to various EBV protein epitopes across the spectrum of diseases that we have here tested in our different groups and control and cohorts. Also, when we are analyzing the viruses, we are not just analyzing virus itself. We also do these structures of the virus, particular parts of the viruses. So we have here in this analysis structure proteins that involved in the viral replications, and proteins associated with different latency programs. So ABV actually can be established through the different forms of latency, like latency 1, latency 2, 2 and 3, and latency 3, each characterized by the expression of a distinct set of viral proteins. In other words, if to simplify these things, it's like you're working with 4 different viruses in the same time, despite they're all called like EBV. So it only makes our situation even harder to distinguish like what is happening what is the reason some people suffer from the long COVID. And what we can see that these latent proteins play a crucial role in viral persistence and host cell manipulation. And that on that heat map, we actually are trying to represent that distinct patterns of antibody responses to these various EBV epitopes are associated with a wide array also of clinical conditions, of chronic conditions. Those can be just like Parkinson's disease, this multiple sclerosis that we already mentioned, type 1 diabetes, some complications of infectious diseases, also cardiovascular diseases, cancers, and so on. So it's like the story, we're only in the beginning of the story, honestly, to uncover the whole mechanism of how the long COVID can actually develop under this circumstance. So we need to understand what kind of conditions person may have, what kind of viruses is already persists in his or her body, and then understand the severity of the systems, and then compare everything to cluster, to analyze this data, and to see how also age or gender may affect or not affect, in this particular case, the situation. So at some point, we are really hoping to reach this point that we can say that, okay, finally, this is the biomarkers through which we can understand whether the person will develop the long COVID or not. But the journey is very long and hard and tough, and we are not slightly, but we're in the middle of that journey, and really I hope to explain more in the next seminar to show more results. So far, I can only say that having those results that we have right now, this is only in the immune system. The next step will be actually to integrate all those results with the different kind of data sets that we have from our partners, and we are trying to do this in all together, obviously through this consortia that we have here, that we are presenting here today. So we're like immunology, like immune system, is only not the smallest part, but just one part of the very hard pattern of how the long COVID is actually developing inside us. And the presence of EBV, it's very interesting because it's an environmental factor. We're also focusing on the genetics. We want to focus on that, just understanding how this genetics is associated with the effect on immune markers. Metabolism also plays a huge role. The lipidome, obviously, everything, and obviously microbiome is also associated. So, I mean, I'm sorry for not bringing such amazing news and showing how complex this world is, but this is the world we're living in, and let's hope for the best, that we finally will be able to understand the patterns, and finally will be able to find the biomarkers that actually can be a work instrument for the doctors. So, and I just wanted to thank all our partners also that works together with the Protobios, from whom we are having so much different data sets that can be involved into analysis to bring a broader picture for the patients who are suffering from the long disease, and also for the clinicians. So thank you so much for listening. I hope you are not getting very depressed by my talk about how terrible the long COVID is. But yeah, we are still going to continue working on that topic. Thank you so much.